Good morning. This is Trishna Ball, and today I am going to discuss with you regarding a very interesting topic, that is vesicular drug delivery systems. And that is liposomes. There are various types of vesicular drug delivery systems, and liposomes is one of the delivery system. Besides liposomes, other type of vesicular drug delivery systems like neosomes, aquasomes, phytosomes, electrosomes, lots of zomes, vesicular drug delivery systems are present in the market, and lots of pharmaceutical companies are working on this vesicular drug delivery systems. So, according to your syllabus, we will be discussing basically on liposomal drug delivery systems. So, before proceeding more into deep into this topic, I would like to let you know about liposomes, which are small artificial vesicles of spherical shape, and that can be created from cholesterol and natural non-toxic phospholipids. Due to their size and hydrophobic and hydrophilic character, besides biocompatibility issues, liposomes are promising systems that are for drug delivery. Liposomes properties differ considerably with lipid composition, surface charge, size, and methods of preparation. Now, as you can see in this particular diagram, the liposome basically it contains two particular parts. One is the hydrophilic head and one is the hydrophobic tail. So, basically liposome exactly mimics that structure of a phospholipid, where it has a hydrophilic head as well as a hydrophilic tail. Now, this is the structure of a phospholipid. So, this is the hydrophilic head. This hydrophilic head basically it contains various types of substances. It may contain choline, phosphate, glycerol, along with various metallic elements like nitrogen, phosphorus, oxygen, carbon, hydrogen, and the hydrophobic tail basically it contains it is composed of the fatty acids. The fatty acids basically it has the double bond within its structure. Now, how does this liposomes exactly works? Whenever the liposomes or liposomal drug delivery systems come in contact with our biological barrier, the liposomes, as you can see, this is the liposomal delivery system encapsulating the green dots, they are basically the drugs. So, whenever this type of delivery system comes in contact with our biological system, and as I must say here, this is the structure of the biological barrier. Our biological barrier basically it contains lipid bilayer. The lipid bilayer, this is the phospholipid bilayer, all right. And so, as the liposomes come in contact with the biological barrier, immediately after a particular time slot, since the liposomes also has the same composition as that of the biological barrier, so these liposomes they have a tendency to get assimilated within this biological barrier, and thereby they totally distribute themselves, or they totally sacrifice themselves within this biological barrier. They assimilate or they totally get distributed easily within the biological barrier, and as you can see in this structure, the liposomes they get they come into one single phase within the biological barrier, and here the drugs are easily released. So, as here we can say, since the liposomes are prepared from various types of cationic lipids, I must stress here cationic lipids. So, these liposomes, once they come in contact with the biological barrier, they are completely biodegradable, they get assimilated within the biological barrier, and thus there is no issue of any biocompatibility or toxicity issues, and the drug release takes place very easily. And also we can say that if you target or if you attach certain ligands attached to this liposomes, so these liposomes the ligand associated liposomes, they can easily be recognized by the specific receptors present on the identified tissues or the cells, and thus we can achieve targeted drug release even by these liposomes. So, as I have said earlier, liposomes are basically small artificial vesicles of spherical shape that can be created from cholesterol and natural non-toxic phospholipids. Basically, when we talk about the lipids should be basically cationic lipids. Other type of lipids are also used for the preparation of vesicular drug delivery systems. When the lipids are non-ionic, they are known as neosomes. Liposomes are vesicular spherical colloidal particles which are composed of lipid bilayer encapsulating an aqueous core. So, basically this type of delivery system, it is mainly utilized for encapsulating a drug which is totally hydrophilic in nature, so that the hydrophilic drug can be easily able to cross through the lipids bilayer which is present in our body. Due to their size and hydrophobic and hydrophilic character, besides biocompatibility, as I have said earlier, liposomes are promising systems for such targeted drug delivery systems. Liposome properties differ variably with lipid composition, obviously, with the surface charge, size and methods of preparation. The choice of the lipid bilayer components determine the rigidity or the fluidity and the charge of the bilayer. Liposomes are extensively used as carriers for numerous molecules in cosmetic and pharmaceutical industries. Liposomes can trap both hydrophobic and hydrophilic compounds, avoid decomposition of the entrapped combinations and release the entrapped drugs at the designated targets. Now, why we need to go for the liposomes, or we can say the rationale behind the preparation of liposomal drug delivery system. These delivery system provide selective passive targeting to the tumor tissues. They result in increased efficiency and therapeutic index of the drugs. They increase the stability of the drug via encapsulation. Results in reduction in toxicity of the encapsulated agents. Suppose a drug, you want to give a drug which is a cytotoxic agent, for example anti-cancer drug. So, by this type of delivery system we can easily give and we can easily target the drug exactly at the targeted site without getting it accumulated at other non-targeted tissues. They result in site avoidance effect. Obviously, when we talk about the targeted delivery system, so obviously the site avoidance will be done. Improve pharmacokinetic effects like reduced elimination, increased circulation lifetime, flexibility to couple with site specific ligands to achieve active targeting. Now, what are the limitations or the disadvantages of liposomes? Basically, liposomes, they have low solubility, short half-life. Sometimes phospholipid undergo oxidation and hydrolysis like reaction. That's why for the preparation of lipid, always it is necessary to check the rancidification of the lipids which are used for the preparation of such type of delivery systems. Leakage and fusion of encapsulated drugs or molecules may occur in such type of delivery system. So, always after the preparation of the liposomal drug delivery system, it is necessary that to check the encapsulation efficiency of such type of delivery system. Production cost is bit higher than in comparison to other delivery system, because you need very good quality of lipids, and there is also you do not know exactly how much quantity of the drug has been encapsulated, because the estimation of the drug encapsulation from such type of delivery system is bit difficult. And very few formulations are only stable, because basically this liposomal drug delivery system, basically they are W by O type of drug delivery system. It is basically an emulsion. Now, based on the structure parameters, these are the various classification of lipids like multilamellar vesicles. The size range is greater than 0.5 micrometer. Oligolamellar vesicles, the size range is 0.1 to 1 micrometer. Unilamellar vesicles, here it consists of all different sizes. Multivesicular vesicles, the size range is more than 1 micrometer. The unilamellar vesicles, it contains various, since I have said that it contains different type of vesicles. So, there are various types of vesicles like medium unilamellar vesicles, small unilamellar vesicles 20 to 100 nanometer, then giant unilamellar vesicles more than 1 micrometer, and large unilamellar vesicles more than 100 nanometer. When we talk about the small unilamellar vesicles, this type of vesicular drug delivery system has a very specific name, and that come under nanosomes or nano liposomal drug delivery systems. Because when we talk about vesicles, these vesicles basically, as you have said earlier, So this is the vesicles. So depending upon what type of or how many layers of these lipids are basically prepared or formed on the inner delivery system, this basically designates the vesicular. If it contains more than one lipid layer, then it will be designated as multilamellar. If it contains one lipid layer, then it will be designated as single unilamellar vesicle. Now, these are the various categories of liposomal drug delivery system based on their method of preparation, like dehydration rehydration methods, or DRV, liposomal system or vesicles, then REV, that is reverse phase evaporation vesicular systems, then multi-MLV, that is multilamellar vesicular system made by reverse phase evaporation method, then vesicles prepared by extrusion technique vesicles, then frozen and thawed multilamellar vesicle, MLV, always it is abbreviated as your multilamellar vesicles, and stable plurilamellar vesicle, SPLV. Depending upon the composition and applications, we have here conventional liposomes, which are negatively or neutral charge phospholipid and cholesterol, fusogenic liposomes, that is RSVE, reconstituted Sendai vesicles, then pH sensitive liposomes, that basically it contains a phospholipid such as phosphatidylcholine or DOPE with oleic acid. Cationic liposomes, it contains the cationic lipids. Long circulatory liposomes, it has high targeting efficiency and made with cholesterol and 5 to 10% efficiency. Immunoliposomes, which are basically attached with monoclonal antibodies. Now, what are the various factors that actually affect the liposomal preparation methods? The first factor is physiochemical characteristic of the material to be entrapped and those of the liposomal ingredients. The nature of the medium in which the lipid vesicles are dispersed, the effective concentration of the entrapped substance and its potential toxicity. Additional processes involved during application or delivery of the vesicles, optimum size, polydispersity and shelf life of the vesicles for the intended application, batch to batch reproducibility and possibility of large scale production of safe and efficient liposomal production. The liposomal, here I would like to emphasize on one particular thing, that is the liposomal encapsulation technology, that is LET, is the newest drug delivery technology that is basically utilized by medical investigators to transmit the drugs that act as curative promoters to assured body organs. This form of delivery system proposal basically targets the delivery of vital combinations to the body. LET is a method of generating submicroscopic forms called as liposomes, which encapsulate the numerous materials. These liposomes form a barrier around their contents, which is resistant to enzymes in the mouth and stomach, alkaline solutions, digestive juices, bile salts and intestinal flora that are generated in the human body, as well as free radicals. The contents of the liposomes are therefore protected from oxidation and degradation. This protective phospholipid shield or barrier remains undamaged until the contents of the liposomes are delivered to the exact target gland, organ or system where the contents will be utilized. In the next presentation, we will be discussing more details about the liposomes. Till then, thank you.